Hello everyone, and welcome to this year's last-minute crash review for your end-of-course biology exam. This video is for students who are about to take a final course test, EOC, or state exam in biology. That means you might be in 9th or 10th grade right now, but that may vary depending on your school or state. In this video, I'm going to quickly review some really important content that's commonly questioned on the end-of-course biology exams and review some basic skills that might pop up on your test. Remember, this is a fast review, so it won't cover everything you learned in biology, and I do talk fast, so feel free to change the speed on your video player if you need. If you need to go back and do a deeper dive on any of these topics, make sure to check out the other resources on my channel. Let's get started. Very briefly, remember all matter on Earth is made of different elements. Elements are composed of different atoms. And in biology, we really want to focus on molecules called organic compounds. Now, these are special molecules that all contain carbon that are essential for life. And the four main categories are nucleic acids, proteins, carbohydrates, and lipids. So when we look at what they're made of, the monomers, or the basic units of nucleic acids, are nucleotides. The basic monomers of proteins are amino acids. And carbohydrates, the monomers are monosaccharides. And the basic monomers of lipids are often fatty acids and glycerol. Now up here I have the basic structure as well. This is actually a phospholipid, which makes up the cell membrane of all living things, and we'll get to that in a little bit. Now to remember all of these organic compound groups, I like the mnemonic clean later, party now for carbohydrates, lipids, proteins, and nucleic acids. One thing I like to add to this is clean later, party especially now, to remember that enzymes for that E are a type of protein, which is really important. Enzymes are really important biological macromolecules, and they belong in the protein category. Now let's talk about water really quick. Water is the universal solvent. It is polar. That means it's partially positive and partially negative on opposite ends. So some molecules are water loving. We call these hydrophilic, and they interact easily. They're soluble with water. Hydrophobic or water-fearing molecules like oils and fats are insoluble in water. Now, water has special properties. It has cohesion, meaning water is attracted to itself. Adhesion is the attraction of water molecules to other molecules or other things, thinking adding something on. Capillary action is when cohesion and adhesion work together, pulling up against gravity. We can see this in stems, in plants, or in a straw even. And water also has a high surface tension, meaning it's more attracted to itself than the air around it, which allows certain bugs like strider bugs and leaves to float on the surface of water. Now let's get microscopic and go down to the cell. Remember, cells are the basic units of life, so all living things have cells. All cells are surrounded by a cell membrane, which is a phospholipid bilayer. We talked about lipids. All living things contain genetic information, DNA, and all cells have cytoplasm. Now if we look just at the cell membrane, let's zoom in really closely. It is actually a layer of two rows of phospholipids, so our bilayer. The heads of these phospholipids are hydrophilic, so they are able to interact with water easily. The inner parts, the tails here, they're arranging themselves away from the water because they are hydrophobic. Now there's two main types of cells. We have eukaryotic cells, which have a nucleus and membrane-bound organelles like the mitochondria. And then we have prokaryotic cells, which also are surrounded by membrane and also have DNA, but they do not have a nucleus and they do not have membrane-bound organelles. They do have ribosomes, they do have a cytoplasm, but we notice here the DNA, which is the scribbles on the diagram, is just floating freely in the cytoplasm, unlike the DNA in eukaryotic organisms, which is in the nucleus. Now, eukaryotic organisms can be animals, they can be plants, and prokaryotic organisms are mostly bacteria. We have archaea as well. Within our eukaryotic cells, we have different types. For example, animal cells over here, which have a cell membrane, and plant cells, which are also surrounded by a cell wall for extra protection and support. Another key difference between plant and animal cells is the presence of a chloroplast, which is shown right here. Chloroplasts are the location of photosynthesis, which plants perform, animal cells do not. Plant cells also have one rather large vacuole, and animal cells also have a vacuole, but it's not as big, and they may have several different vacuoles as well. And remember, both plant and animal cells are both eukaryotic. Now let's zoom in once again on that cell membrane. Remember, it is made of a phospholipid bilayer. Now different molecules can get in and out of the cell membrane in different ways. We have simple diffusion, which is the movement of particles from a higher concentration to a lower concentration. We have facilitated diffusion, which allows particles to move from a higher concentration to a lower concentration, but across a membrane, facilitated, being helped. This protein channel is helping the molecules get through the membrane. Then we have active transport, which allows molecules to move from a low concentration to a higher concentration with the help of energy in the form of ATP. So a way I like to remember this is kids going down the slide. So simple diffusion requires no energy. It's just a kid going down the slide from high to low by themselves. Facilitated diffusion might have a parent or a friend, which is representative of our protein, and they're going from high to low as well, still requiring no energy themselves. And then active transport, think of a child going from low to high, going up the slide, so against the concentration gradient, but they have to use energy to do that, so they're using ATP. All right, when water moves across a membrane, this is called osmosis. This is water moving from a higher concentration of water particles to a lower concentration of water. And when we think about where the water is moving in comparison to the solute or the other particles dissolved in the water solution, here we have kind of an equal balance of particles in and out of the cell that are not water, so we would call this an isotonic solution, and water would move freely in both directions in about the same amounts. In this situation, we have a higher concentration of particles or solute inside the cell, but a higher concentration of water particles, which is the blue here, outside of the cell. So where would the water move? The water would move into the cell. This is a situation called a hypotonic solution, where there are fewer solute particles outside the cell, more inside the cell, more water outside the cell, a lower concentration of water inside the cell, so water would move in. And now here we have more solute particles outside the cell, or a higher concentration of solute particles. So remember, where is water going to move? From a high concentration of water, so that means it would move outside the cell. This is called a hypertonic environment. Now, in a hypertonic environment, there is a chance that if a lot of water moves out of the cell could actually shrink, and the opposite would happen in a hypotonic environment. A lot of water may move into the cell, causing it to swell or burst. Let's get back to enzymes. Remember, these are a type of proteins. They are extremely important to living things. They are biological catalysts, meaning they get things going, they get reactions started. Things like ligase, helicase, polymerase, amylase, these are all enzymes. Notice all of these words end in the letters ASE, which makes it easy to recognize an enzyme even if you've never seen it before. Some things that can affect enzymes are temperature or pH. If enzymes get too hot, they can denature or unfold, and then they'd be unable to act with their substrate because they no longer have the shape of the active site, which is where a protein or an enzyme usually interacts with the substrate, the molecule that it's going to do something to or interact with. All right, let's jump over to cellular respiration. Now, all living things need energy, and one of the main ways they get that is through cellular respiration, which occurs in the mitochondria. This is the process where organisms take glucose, sugar, and oxygen and go through reactions to transform it into ATP energy. In that process, carbon dioxide and water are also generated. So if we think about this on a larger scale, when we eat, we consume glucose. When we inhale, we take in oxygen. And when we exhale, that CO2 is a byproduct of cellular respiration. We exhale water vapor as well. You can see that coming out of your mouth on a cold day. And then ATP is also generated, which gives us the energy to perform our daily activities and survive. All eukaryotic organisms are going to perform some type of respiration. Another important reaction we need to be aware of is photosynthesis. This is how plants get the food they need, and they use sunlight energy to convert the carbon and carbon dioxide into food molecules like glucose. So carbon dioxide and the sunlight energy in water can be transformed into glucose and oxygen. Remember that plants need that glucose and oxygen to perform cellular respiration themselves. So these processes are complementary and they feed each other, which is why photosynthesis is crucial to life on Earth. Another type of respiration is fermentation. This is often called anaerobic respiration. It's a type of anaerobic respiration, but that means it occurs without oxygen. So bacteria, some yeast perform this process, and fermentation is not as efficient as cellular respiration. Cellular respiration produces 36 molecules of ATP, where fermentation only produces 2 molecules of ATP. So even though we can also perform fermentation in our muscle cells, for example, when we run out of oxygen, cellular respiration is a much more efficient process for producing producing energy molecules. So like I said, the type of fermentation that we can do in our muscle cells is called lactic acid fermentation, and that's because we take glucose and we generate energy molecules, ATP, but also lactic acid as a byproduct. Now, yeast can do another type of fermentation called alcoholic fermentation, and here the yeast produce ethanol as a byproduct or a type of alcohol and carbon dioxide. So that carbon dioxide we can see as bubbles forming if you've ever done an experiment with yeast and fermentation. It's why bakers use yeast in their baking, because that carbon dioxide can help bread rise. And then also, of course, ATP is generated in this process. Let's get down to the DNA in our cells. Remember, all living things contain DNA. Now, DNA is organized. It is wound up into chromosomes and condenses into chromosomal form before the cell can divide. Remember, each double-sided chromosome that looks like this actually is a duplicated set of DNA. So we have two sister chromatids here. They're connected at the center by a centromere. But usually within the cell, if the cell is not preparing for cell division, it may not be condensed into these chromosome forms. It may just be in chromatin form, which sounds similar to chromosome, but it kind of is represented as this like spaghetti-like drawing where the DNA is unwound. It's not condensed. As the cell prepares for cell division, we may see the chromosomes begin to look like this. That means the DNA has already been duplicated and the chromosomes are arranging themselves so they can prepare for cell division. Mitosis is a type of asexual reproduction where cells make exact copies of themselves. One cell can generate two identical daughter cells. You might have learned the phases of mitosis as PMAT, which stands for prophase, metaphase, anaphase, and telophase. And then at the end, there's cytokinesis, where the cytoplasm actually divides. Now in prophase, the nuclear envelope dissolves and the chromosomes condense. They start to move to the middle of the cell. In metaphase, they align along that cellular equator or the center of the cell, and these spindle fibers on opposite ends of the cell come and attach at the centromeres. In anaphase, think A for away, the sister chromatids are separated. They start to get pulled away from each other towards opposite ends of the cell. In telophase, new nuclear envelopes start to form. The cell starts to separate, and in cytokinesis, we have the exact separation of the cytoplasm. Now with mitosis, you've probably also studied meiosis, which is a different type of cell division. Mitosis, remember, will copy body cells, make exact duplicates of cells. So one parent cell will create two identical daughter cells, whereas meiosis is the process used to generate sex cells. So one parent cell that goes through meiosis will generate four daughter cells, but they'll only have half the genetic information as the parent cell. So meiosis takes a little bit longer. It goes through PMAT twice, and you don't need to memorize what happens in every single stage, but the results are important. So at the end of meiosis, there are going to be four daughter cells, each with half the genetic information as the parent cell. And this genetic information has been mixed up a little bit, so it's a different combination in each cell. So the genetic information is different at the end of meiosis, which is going to be helpful in the diversity of life on Earth as organisms reproduce sexually. Now, as organisms reproduce, we pass on our genetic traits. Gregor Mendel was often called the father of genetics. He did studies that recognized that a lot of traits are controlled by two different alleles or versions of a gene, and alleles can be dominant or recessive, meaning one can cover up a particular trait. So we have these words, heterozygous, homozygous, to talk about genotypes or combinations of genes. So for example, this big T would be a homozygous dominant genotype, and each of these letters represents a version of that gene. So you could pause and try to fill this in for yourself, but here we go. We have big T would be homozygous dominant. Big B, little b would be heterozygous because it's two different versions of the same gene, and little t is homozygous recessive, two different versions of the recessive gene. Remember, a big T often represents a dominant allele, and a little or a lowercase letter would represent a recessive allele. So for example, if yellow body color is dominant to blue in these fish, if we have a big Y and a little y, that would result in a fish being yellow. That's the phenotype. So if we write the phenotypes here, we would see what would happen with each of these genotypes. Remember, genotypes are the combination of genes. Phenotypes are the actual physical characteristics that we get from the genotype. So you can pause this and go through each of the examples if you want. And you might have also practiced Punnett squares in biology, where you have to separate the alleles of both parents on this table and figure out the possible combinations that could result from this cross, or from when these parents mate, what their offspring could potentially have. And remember, what happens in a Punnett square is just a probability. It is a potential outcome. We won't know for sure until the offspring are born what the actual genotypes and phenotypes are. And remember, most human traits, most genetics are very complicated, so it's not as simple as Gregor Mendel said. There's different types of dominance, like incomplete dominance, where there's a blending of traits, codominance, where we see representations of both traits. So for example, if a purple and a pink flower were codominant for flower color, we might see both purple and pink petals in their flowers. And of course, there's also sex-linked traits, which are traits that are carried on the X and the Y chromosomes. Now, humans have 23 pairs of chromosomes. That 23rd pair is going to be the sex chromosomes, and females have 2 X chromosomes, males have an X and a Y. When we talk about traits like color blindness, these are traits that are carried on the chromosomes that are sex chromosomes. So when we perform genetic crosses with sex-linked traits, we'll also write the X and the Y there and the allele as an exponent, and make sure we carry all of the letters together in our Punnett square crosses. Now, genes can also be influenced by the environment. For example, these hydrangeas can have different colors based on the pH of the soil, and this can go for a lot of different traits. Sometimes there's temperature differences that can change the color of fur of different mammals. And of course, chromosomes can have problems separating during meiosis or mitosis. And when we have that non-disjunction, or those sister chromatids not going to the opposite end of the cell correctly, we could end up with a cell with different numbers of chromosomes. For example, trisomy 21 would mean that we have 3 copies of the 21st chromosome. And trisomy 21 is also known as Down syndrome. And that means that person would have a condition that affects their physical characteristics, some of their development, because of that extra copy of that chromosome. Now let's talk about DNA a little bit closer now. Remember that in DNA we have 4 different bases: A's, T's, G's, and C's. A's always pair with T, G is always pair with C. DNA replicates during the S phase of the cell cycle. And in DNA replication, we have the DNA strand separating, and new strands of DNA are built off the template. So it's called semi-conservative replication. Each new strand of DNA has half the old strand and half brand new. If we look back at our organic compound structure, each nucleotide has a phosphate, a sugar, and a base, A, T, G, or C. And so when we put these together, A's and T's, we can see always pair together, where these are little hydrogen bonds in the middle. G's and C's always pair together. And the backbone of the DNA molecule is made of phosphate and sugar molecules. So how do we get exactly from our DNA to our actual trait? Well, the process is called protein synthesis. And in the nucleus, where the DNA is, the DNA will separate, and then an mRNA sequence will be built off of the DNA template. That mRNA will leave the nucleus, and that process is transcription. Then we'll go through translation, where the mRNA will go to a ribosome, and tRNA molecules, a different type of RNA, will match up with the codons, or the different groups of mRNA, and bring over a matching amino acid. And then those amino acids will start to link up. And remember, amino acids make proteins, and so that is how we deliver the message to build the protein that is going to give us some trait in our bodies, like the protein that influences eye color, or the protein that gives us our hair shape. So some differences to remember between DNA and RNA. DNA is double-stranded, RNA is single-stranded. DNA has actually a deoxyribose sugar, that's where its name comes from, deoxyribonucleic acid, where RNA is made of a ribose sugar. And then DNA's bases are A's, T's, G's, and C's, where RNA actually has a U instead of a T. So if you're creating an RNA strand based off DNA, remember to replace wherever a T would go with a U. Let's do a little practice. So if we're transcribing a sequence of DNA, this purple is the DNA here. The sequence is A, G, T, G, T, C. The complementary RNA would be A pairs with U, G pairs with C, T pairs with A, G pairs with C, T pairs with A, and C pairs with G. So again, try this on your own. Go ahead and pause real quick. The correct mRNA sequence here would be AUG, CGU, ACU. And then don't forget the next step is translation, where we build that protein. And to do this by hand, you might see a codon chart where you're asked to find the codon and then the matching amino acid. So let's say we were looking at a codon that was CCC. So we'd start in the center, find the C, then find the next C and the next circle, and then go to the final circle here, find that C that refers to pro, which is short for proline, one of our amino acids. You do not need to memorize all your amino acids, and if you're ever asked to do translation on a biology exam, you will be provided with a codon chart. Codon charts can also look like this. So let's look at CCC again. C is our first base here. We would go to the top for our second base. Here's another C, and then find that third base C that also corresponds to proline on this chart. It is a universal genetic code. Let's talk really quickly about biotechnology. Now, there's lots of different growing and emerging new technologies in the field of biotechnology, from lab-grown meat to DNA tests to genetically engineered organisms to resurrecting woolly mammoths to CRISPR DNA. And so, of course, there's ethical questions that go along with all of these new techniques. One technique you might see on your exam would be gel electrophoresis, which is a way to identify different parts of DNA. So gel electrophoresis can be used to study evolutionary relationships, identify DNA found at crime scenes, or maternity or paternity testing. So here we have a simple gel. In the gel, DNA is loaded at these wells at the top. DNA is negatively charged, so it'll be attracted towards the positive end. And then these DNA samples will travel down this gel as we run an electric current through the gel, and we can see which pairs match up. So here we have a mother which matches with child A and child B. Child C, though, does not have any matches with the mother, so we can say that child C is not related to this particular mother. Now, on a much larger scale, remember that evolution is the change in genetic makeup of a population over time. Natural selection is the major mechanism behind evolution, and when we talk about an organism being evolutionarily fit, that means how well they are able to survive and reproduce, not how strong they are, not how fast they are. It's really about how they can survive and pass on their traits to their offspring. And remember that environments are a huge factor in evolution, and they can act as a selective pressure, and different organisms will have better rates of survival in different types of environments. An adaptation is an inherited trait that is favorable to an organism, that helps an organism survive. And Charles Darwin was one of our main scientists that created this idea of natural selection. We have lots of evidence now to support this idea of evolution, from the fossil record to biochemical and DNA evidence, embryology, and we can see how different organisms develop very similarly through the study of their embryos. A phylogenetic tree, or a cladogram, is a way to look at different evolutionary relationships over time. And when we see branches on a tree, we can see where their last common ancestor might have been, and we can also add traits to this tree. So if we look at a tree like this, we can read it as anytime there's a trait that appears, any organism that comes after that trait developed that trait. So for example, the organisms on this tree that have vascular tissue are ferns, pine trees, and flowering plants, but not mosses. And the organisms, or the groups of organisms that have seeds, are pine trees and flowering plants, but not ferns and mosses. Now, backing all the way up to the biosphere, remember different levels of organization in biology go from molecule all the way up to all life on Earth. And at the population, community, and ecosystem level, we can talk about biodiversity, which is a measure of the variety of different types of organisms in an ecosystem. And biodiversity is very important to living things, because the more biodiverse an ecosystem is, the more resilient it will be. Now, of course, environments are always changing. Large-scale changes that can influence ecosystem can be maybe the addition of different nutrients to a system. We could have forest fires, we could have droughts, we could have floods, earthquakes. There's so many things that could influence and create major changes to an ecosystem. In a food chain, we can see that organisms that produce their food often come first. So producers, like these autotrophs or grass, are here. And we always draw the arrow pointing towards the organism that is doing the eating. So think about this food going into this elk's stomach. That's the direction of arrows for a food chain or a food web. Now we have our producers, or our autotrophs here. Our heterotrophs are organisms that get their food from something else, so they don't make it on their own, they consume their food. This is also a heterotroph, and this is also a heterotroph. And when we go up a food chain or a food web, we can also identify organisms as producers or consumers. So this is a producer. This is a primary consumer, because it's the first organism that eats the producer. This is a secondary consumer, and this is a tertiary consumer if the human were to eat the bobcat. On a trophic pyramid, remember that the most energy is found at the bottom of the pyramid, and as you go up a level, most of that energy is lost to the environment. So it's more efficient to consume lower on a trophic pyramid if you're an organism. There are tons of different molecules and atoms cycling throughout our environment. Carbon is one of them, and the carbon cycle involves both photosynthesis, the plants harnessing sunlight energy in order to take the carbon dioxide from the air and create glucose molecules, respiration, we take that glucose and we use it to create energy energy and then we exhale the carbon dioxide. Decomposition can break down carbon in organisms' bodies, and combustion also releases carbon dioxide into the environment. Now, there are tons of resources on Earth. Some are renewable, which means they can be replaced quickly by natural processes. Some are non-renewable, meaning they cannot be replaced generally within human lifetimes or as fast as they are being used. And so we need to consider our use of resources as the population of humans continues to boom on planet Earth. A hole in the ozone layer is a problem that we've identified recently, but it's actually getting better. But this hole produced by the use of CFCs, or a chemical called chlorofluorocarbons, was pretty dangerous because it was letting in harmful UV radiation into the Earth. Now, this is separate from the issue of global warming, which is predominantly affected by the combustion or burning of fossil fuels, because that adds gases to the atmosphere that cause it to hold more heat, and continued global warming can lead to rising sea levels, coastal flooding. But there are other human impacts, plenty of human impacts on the environment, such as introduction of invasive species, introduction of diseases, changing habitats, deforestation, industrialization, all of these things. But humans can also have a positive impact on the environment by reducing our consumption of resources, recycling, planting native species, protecting wildlife and habitats, and supporting sustainable practices. Whew, we're coming to the end. I hope this has been helpful as you cram for your biology exam. I have lots of other biology, AP biology, and other life science resources on this channel, so be sure to subscribe so you don't miss out. Best of luck on your biology exam. Let me know if you have any more questions in the comments below. Thanks so much for watching, and give this video a like if it's been helpful. I'll see you later.